Okay, everybody, we are going to move on to chapter 2, the chemical basis of life. We're going to be a little selective in this chapter, so you do not need to read the entire chapter. You have 2 select readings in D2L again. That will take you into Connect for the ebook, but we're pretty much going to skip over section 2.1 and section 2.2. So we are going to focus on acids and bases, the concept of acids and bases. And we're also going to talk about inorganic and organic molecules. So we will not go over the entire chapter 2, but this is the most relevant stuff when it comes down to the chemistry side of things that we need to know to understand our physiology. Okay, we talked a little bit about homeostasis last time, and one of the examples or systems that needs to be maintained is our balance of acids and bases. So acid itself is considered a proton donor or an H plus donor. This is a hydrogen here. And so hydrogens are basically just one proton. H plus, it's just one proton, so it has a positive charge. And what it means by a proton donor is that, we'll use our example of hydrochloric acid here, is that if we put hydrochloric acid into a solution, it is going to try to give away its hydrogens. It's going to dissociate into hydrogens and chloride. Now a base, on the other hand, is going to be a hydrogen acceptor or a proton acceptor. And so when we put, in this case, sodium hydroxide into a solution, it is going to split into sodium ions and a hydroxide ion. That hydroxide ion, or what you'll see sometimes written as an OH negative, that can accept the H plus, that can accept our proton. So if we were to put hydrochloric acid and sodium hydroxide together, that would result in sodium chloride or NaCl salt and water, because the sodium, positive sodium ion and the negative chlorine ion would combine with one another, and our hydrochloric acid would donate its hydrogen to our hydroxide acceptor. So you would get NaCl and H2O, so salt and water, if we were to mix these two in particular. So you can have different strengths of acids and bases, and what makes one stronger than the other is the amount it dissociates. So for example, hydrochloric acid is a very strong acid. So if I put that into a solution, it's pretty much all going to split into hydrogens and chlorine. So we're not going to have any that remain as that HCl or hydrochloric acid still. Same thing with sodium hydroxide. It's a very strong base. So when I put it into solution, it's going to dissociate into its sodiums and its hydroxide ions. If I had a weak acid or a weak base, only a few of them would dissociate or separate to be a donor or acceptor. So when we're talking about acids and bases in the body, you'll mainly hear this referred to in the term pH. So pure water, and there's the next slide has kind of the scale to see a little better. Pure water is going to be exactly at 7.0. So that's neutral. Exactly 7 is pure water. It's not acidic or basic. Anything below 7 is going to be acidic. Anything above 7 is going to be basic. So our scale goes from 0 to 14. So don't worry too much about the concentration here. This just means at the bottom part here, we're not going to have many protons. And then with our strong acids like hydrochloric acid that has a pH of 0, meaning it's super acidic, that it's going to have a high dissociation, or we're going to have a lot of protons or H pluses in our solution. So again, if we look at here, distilled water, we're at exactly 7.0. As we go down, you're going to continue to get more acidic. So the closer to 0 you get, the stronger the acid is. So like you see stomach acid and pH of 0 for our hydrochloric acid. Now, if we go the opposite direction, we're going to be going to stronger bases. So if you look all the way up top here at 14, we have sodium hydroxide. So that's the strongest base. And so you'll hear the term basic, but you'll also hear the term alkaline, something that's really popular in kind of the health field right now, or even social media. You'll see alkaline water is being sold to help decrease acidity in the body. So that's another term for basic, is alkaline, or alkalinity versus acidity. So if you see blood here, that's where we are concerned. Normal is about 7.4. So in our homeostasis conversation from last chapter, we talked about a set point as well as a normal range. So 7.4 would be our set point, and our normal range for the most part is going to be about 7.35 to 7.45. We want to stay as close to 7.4 as possible, but all our functions are going to be relatively normal as long as we stay within that 7.35 to 7.45 range. That's not very much wiggle room, so we constantly have to be trying to balance the acids and bases, because it doesn't take a lot to throw our pH out of whack. So what you guys need to remember is that distilled water is going to be exactly 7. That's neutral. Make sure you know that our scale goes from 0 to 14, less than 7, more acidic, greater than 7, more basic or more alkaline. And so the closer to 14 we get, the stronger base or the more alkaline something is going to be. And the closer to 0 we get, the more acidic something is going to be. Okay, we're going to move on to inorganic versus organic. And so really the big distinction between inorganic and organic is the presence of carbon. So something that is inorganic or inorganic chemistry is going to deal with substances that do not contain carbon, do not contain carbon. So vice versa, our organic chemistry is going to be carbon-containing substances. So inorganic means no carbon is present. Organic, carbon is going to be present. So there is a slight exception to the rule. There are some carbon-containing compounds that are not organic or considered inorganic because they do not also contain hydrogen. That'll make a little bit more sense when we start discussing some of our organic molecules. You'll see that not only is there carbon, but there's also hydrogens. Okay, so let's discuss oxygen and carbon dioxide first. So oxygen is quite a small molecule. So we have oxygen on the periodic table is a single O symbol. But when we're discussing about oxygen in terms of the air we breathe, we're talking about O2. It's small, it's nonpolar, meaning it's not charged, and it's inorganic because it does not contain carbon. So oxygen is 2 oxygen atoms that are bound together by a double covalent bond. Now, you guys don't have to worry too much about the different types of bonds. That's something that's more on the chemistry side of things. But there's a few different types of bonds that we'll talk about, and it just changes somewhat the charge or the polarity of different molecules depending on how they're bonded together. So of course, we all know oxygen is essential for most living organisms. And on the flip side, we have carbon dioxide. So that's when we have a carbon that's bonded with 2 oxygen atoms. And typically for us animals or humans, when we break down food molecules and metabolize them, carbon dioxide is going to be one of our waste products. So that's one carbon atom and two oxygen atoms. And remember, this is still considered inorganic in this case. Even though there's that carbon there, we have no hydrogens. So both oxygen and carbon dioxide are going to be considered inorganic molecules. Okay, water is also an inorganic molecule. So we have some hydrogens there, but we have no carbons. So we have two hydrogens that are bound by polar covalent bonds, meaning there's a charge associated with them, to an oxygen atom. And really, these hydrogens are bound by what's known more specifically as hydrogen bonds. And that gives water a lot of its properties, or what makes water so important and how it interacts is the way the hydrogens and oxygens are bound together. It kind of allows it to stick to other water molecules in a special way. So the two terms that we're going to discuss here are hydrophilic and hydrophobic. So it's going to be attracted to some other polar molecules. Think kind of opposites attract. Positives are going to be attracted to negatives. Negatives are going to be attracted to positives in terms of charged molecules. So any molecules that are attracted to water are going to be known as hydrophilic. So hydro meaning water, philic meaning love. So they love water. They're attracted to water. The opposite of this is going to be hydrophobic. So think, if you've ever heard the term phobia, meaning fear. So they are fearful of water. They're not going to be attracted to water. So some things love to be in contact with water. Some things avoid water at all costs. If you've ever tried to mix oil and water together, that's kind of a good visual for hydrophobic. They don't want to interact. So the oil is hydrophobic. So water performs a lot of essential functions in the body, but we kind of summarize this in three big ones. We have the stabilization of body temperature. It's actually protective for the body, and it's going to be required for many of our different chemical reactions. So stabilized body temperature, protection, and needed for chemical reactions. That's our three essential functions. Okay, now we're moving on to organic molecules. So carbon's ability to form these covalent bonds with other atoms makes it kind of one of the largest building blocks for many different diverse, very large, complicated molecules that are essential for life. Really, this is kind of, I know we're zoomed in now in terms of looking at it on a very small scale. These molecules, even though they're large and diverse, are not large as if we were looking at them, but very important. So we're going to talk about four major groups of these organic molecules: carbohydrates, lipids, proteins, and nucleic acids. So these are our four groups of organic molecules. Okay, we're going to discuss carbohydrates first. Excuse me, guys, a sneeze. Okay, so carbohydrates are going to contain carbon, hydrogen, and oxygen. Hydrogen to oxygen ratio is going to always be 2:1 in carbohydrates. Not always in organic molecules, but specifically referring to carbohydrates. So the example they show here is glucose. So C6, 6 carbons, H12, we have 12 hydrogens. So that means we're going to have 6 oxygens. So twice as many hydrogens as we have oxygens. Okay, so when we talk about carbohydrates, we classify them by the amount of sugars that exist. So our monosaccharides are the most simple form of our carbs. They just have one sugar. So examples here are glucose and fructose. You see there's just one carbon ring. one carbon ring. So they're just one sugar. So there are building blocks. We can put together our monosaccharides to create our disaccharides or our polysaccharides. So our disaccharides are going to be two sugar chains. So if we added glucose and fructose, two of our monosaccharides, we would get sucrose, which is a disaccharide. Another example of this, if we took the monosaccharide glucose and the monosaccharide galactose and have them combined, they're going to be a disaccharide known as lactose, found in many dairy products especially. And then polysaccharides could be many sugars. So something that has 3 sugars is a polysaccharide. Something that has 3,000 sugars would be a polysaccharide. Once you get above 1 and 2, then you fall into this polysaccharide category. So this is what we usually would think of when we're talking about carbohydrates. So starches are grains. Vegetables tend to have a fair bit of carbohydrates in them, not compared to our starches and grains, but more than anything else. And glycogen, remember that's our stored form of glucose, is glycogen. So when you see the term carbohydrates, the first thing you should think of is energy. So carbs are our main energy source. So we also use carbohydrates to provide parts of other organic molecules. One example of this is our nucleotides in DNA. So it can make up some of our other organic molecules. I said the big one that you should always think of immediately is that carbs are going to be what we can break down to provide energy. And if we don't digest carbs, they end up providing bulk, or they become our fiber in our feces. So that's how we eliminate excess or undigested carbs, is kind of through the form of or with fiber in our feces. Okay, like I said a little bit ago, glycogen is going to be our main stored form of glucose in humans. It's going to be considered a polysaccharide because it's a lot of glucose molecules stacked together. So when we need this, we have these stores both in our muscles as well as our liver. And so we can break these stores down to make them available to our cells, and our cells can utilize our glucose or our carbs to make ATP. And we'll talk about ATP in this lecture a little bit later, but ATP is really our main energy source. It's the actual energy that our muscles are using. So we utilize carbohydrates to create ATP. That's how we get energy from carbs. Starch and cellulose are important polysaccharides that are found in plants. And humans will ingest starch when we eat lettuce, broccoli, whatever it is. Then we are going to be able to use the carbohydrates or the polysaccharides in those foods, just like we would if we ate pure sugar, to be able to make ATP. Humans cannot digest cellulose, but it does provide a lot of fiber, which we need in our diets to help us eliminate our excess carbohydrates. So starches we can break down to utilize for energy. Cellulose, we're only getting fiber from. We cannot use cellulose to make ATP. Okay, our next category is lipids. So another word for lipids would be kind of like fats. It's a little bit more involved than just fats. Fats are really just one category of lipids. But when we're talking about diet especially, you'll hear this grouped as kind of the fat category. So this is also going to contain carbon, hydrogen, and oxygen. It doesn't have as many oxygens to carbon as carbohydrates do. These are typically going to be hydrophobic, meaning insoluble in water. Remember, we talked about that oil in water example for hydrophobic. So oil is a lipid. Any fats, cholesterol falls into this category. You often hear in terms of medication or in the medical side of things, people are talking about high cholesterol, but cholesterol has a lot of important functions as well. So we need cholesterol, triglycerides, and phospholipids. So different categories of lipids serve different functions. So we'll talk about triglycerides first. And these are really our dietary fats, is what you should think of triglycerides as. They provide really good long-term energy storage. So our glycogen and our glucose, those are more short-term and kind of easily accessible, easy to process. Now our triglycerides are going to be a longer-term energy storage. So we use fat for energy as well, but it's not as quickly available as our sugars are or our carbs are. Of course, fat provides insulation so that we don't lose heat, and is also protective cushion for organs. So our adipose tissue or our fat tissue is going to provide some cushion and protection. Cholesterol is a very specialized lipid. They're part of our cell membrane structure, so they're important for structure, and they have many other functions in the body as well, but we're not going to get into details on that. Phospholipids are also specialized lipids, and their main job is to compose our cell membrane. So cholesterol provides us some cell membrane structure, and a majority of our cell membrane structure is provided by phospholipids. So let's talk a little bit more about triglycerides. We can break these down further into a saturated group and an unsaturated group. And what saturated means is that we have single covalent bonds between our carbon atoms. And saturated fats are typically going to be the ones that you think of as solid at room temperature. So your meat, fat, beef, and pork, butter would be considered a saturated fat. Cheese falls into this category. Eggs versus unsaturated, we're going to have at least one, if not more, double covalent bonds between carbons. So saturated, think S saturated, S single. So single bonds when it's saturated, multiple double covalent bonds, or at least one double covalent bond if we're unsaturated. So typically you think of unsaturated as our oils, olive oil, fish oil, sunflower oil, canola oil. All of those ones that are going to be liquid at room temperature are typically going to be unsaturated. This just shows a little bit of an example of our actual triglyceride structure. So we have a glycerol molecule here. Don't get too caught up in this slide, just to have an example, with 3 different fatty acids. And then this shows we have some enzymes that come into play to convert this glycerol and fatty acid molecule. We lose 3 water molecules. You'll see our HOH, HOH, HOH. We lose those as water molecules. And now we have our oxygen bonded to the carbon. So our triglyceride, we have 3 fatty acids that are bonded to a glycerol. So this is our phospholipid structure here. You see, along with our oxygen, carbon, and hydrogen, it also contains a phosphorus. So that's where the phospho comes from. And it also has some nitrogen. And so we talked about the terms hydrophilic and hydrophobic. So our phospholipids have both of these properties depending on which portion we're talking about. So we have a head and tail. So our head is going to be hydrophilic. This is our polar region. It's got a charge to it. It likes water. And then we have our tail, which is hydrophobic. This is where our fatty acids are contained. So our phosphate region is the head. That's hydrophilic. Our fatty acids are our tail, and they're hydrophobic. Okay. So what you see in the cell membrane is that it's a double layer of these phospholipids. So you'll have one layer all stacked together, kind of one after the other, where we're going to have the polar hydrophilic heads facing the external environment of the cell. And then we're also going to have another layer where they're kind of tail to tail. So our heads are going to point towards our external environment and the internal environment. And then our tails are going to be stacked and they form the middle portion of our cell membrane. Okay, proteins. So these are going to contain carbon, hydrogen, oxygen, and nitrogen. So when we're discussing proteins, our building blocks are going to be amino acids. So amino acids are the building blocks of our proteins. So we have 20 different naturally occurring amino acids. Some are considered essential versus non-essential, meaning that our essential ones we have to eat to be able to get them into our body. The non-essential ones our body can produce or create itself, but our essential amino acids we have to eat. So they're not always just stored. So we need to continue to eat protein to be able to replenish our amino acid building block storage. We need to bring those back in as we use them up. And we don't talk about this too much here, but after we utilize a protein, oftentimes we can then break it back down into its building blocks so we can use those amino acids again to make the same protein or for a different protein. So sometimes our proteins and amino acids kind of get recycled to use them again, but we do always need to replenish our supply. So that's why protein is a key component in our diet. And so proteins are probably the most important organic molecule because we use them for absolutely everything. They are essential to all of our cellular functions and structure. So they make up our skin, our hair, our nails, our muscles. They're part of the hemoglobin molecule, which is how our red blood cells transport oxygen to cells and bring carbon dioxide back out of cells. They act as enzymes. And when you think of enzymes, we'll talk about them a little bit more, but basically they're essential for a lot of our cell processes. They don't happen unless they have an enzyme to assist. Our proteins are essential to our immune system function. Of course, actual muscle contraction. So actin and myosin are two big proteins or important proteins that we'll discuss in our muscle chapter when we're talking about muscle contraction. And these also make up part of our cell membrane. They have some different jobs. There's some different types of proteins along our cell membrane. Okay, protein denaturation is when we break down our proteins and it becomes non-functional. So what occurs is that the hydrogen bonds that are throughout, so tons of different hydrogens you see in there bonded, they kind of help maintain the structure of our proteins. And if those become broken or not as reliable, our protein is now going to be non-functional because it needs that structure to actually do its job. So typically, what causes this process is either too high of a temperature. That's part of the reason why a super high fever can be a real problem if it gets to a certain point where it's going to start to denature our proteins. And this goes back to pH that we talked about earlier. We have to maintain that 7.35 to 7.45 range, because if we fall either higher or lower than that, our proteins are not going to be in the right environment to function. And you see they do so many things in our body, so that becomes a big problem very quickly. Okay, so we'll talk about enzymes here. I was saying a bit earlier that they're essential for a lot of our functions, and this shows why a little bit. So they're considered a catalyst, meaning that they speed up the rate at which our biochemical reactions or our life reactions happen at. So enzymes are organic catalysts. They increase the rate that our reactions proceed without that enzyme being permanently changed. So why this is important is because enzymes allow lowering of our activation energy needed to start the reaction. So let's imagine that this protein or this enzyme was not here. And for example, let's say it takes 10 ATP to combine molecule A and B into AB. But if we add our enzyme, now let's say it only takes 5 ATP to make this happen. So we're significantly lowering that activation energy to allow our process to happen without having to use up so much energy. These are happening all over the body. So if we needed to have a lot of energy available all the time without enzymes, we'd probably run out. So enzymes allow us to use much less energy. It makes us much more efficient, much quicker at doing all of these different reactions that need to happen. So you'll see here, we have our red molecule A, our blue molecule B. It'll have a very specific enzyme for this reaction. So it'll be shaped so that molecule A can fit right on one arm and molecule B can fit right on the other. That enzyme assists in speeding up that reaction to create molecule AB. And after that reaction happens, you can see our enzyme is unchanged. So it changes shape in this middle part to help connect our two molecules here. But at the end, even though we have AB now, our enzyme looks exactly the same as where we started. Okay, last category is nucleic acids. So again, of course, we're going to have carbon, hydrogen, and oxygen. We're also going to have nitrogen and phosphorus. So you'll see our two big examples are DNA and RNA. This example shows DNA here. And so in your reading, you can look a little bit more about the details. I'm not gonna read this word for word to you guys. But our nucleotides are the building blocks in this case. So for both DNA and RNA, we have 4 different nucleotides. So this example is DNA. You have cytosine, guanine, thymine, and adenine. So they always partner with each other. So you'll always see adenine and thymine together here, as well as cytosine and guanine. And so in RNA, the only difference is rather than thymine, rather than seeing these T nucleotides, you're going to see, or bases really, or the whole structure is a nucleotide. Our letters where we're colored here, these are really just the nitrogen base. This T is going to be a U. So RNA has uracil, or a U, instead of our thymine. So the nucleotide is composed of that base, as well as our 5-carbon sugar here, and a phosphate group. So that's what makes up our nucleotide. So we have the nitrogen base, our sugar, it's a deoxyribose, so that's why it's called deoxyribonucleic acid, and our phosphate group. So one other thing I can point out here is that you see DNA has what's known as this double helix structure, or it's double-stranded, where RNA is just going to be a single strand. But we'll get more into that in a later chapter. Okay, ATP. We talked about this earlier in terms of ATP is our energy. So sometimes you'll hear it referred to as the energy currency of cells because it's capable of both storing energy and providing energy. So when we put these phosphate groups together, these bonds store energy. When we break one of these bonds, that's going to release energy. So you'll see here it consists of our adenosine or our adenine base. So adenosine is our sugar, our ribose sugar, and our base adenine. So that's our adenosine. If you kind of follow this, we have adenosine is just this section. So our ribose and our adenine, that's adenosine. When we add on our phosphate groups, we change the name. So if we cut it off at this kind of our adenosine plus 2 phosphate groups, we have adenosine diphosphate, ADP, and we typically are referring to adenosine triphosphate. So 3 phosphate groups. We have our adenosine plus 1, 2, 3 phosphate groups. Okay, I don't want to get too much into the weeds on this chapter. Like I said, this is another building block chapter that'll help us to understand our physiological processes as we go kind of within each system. Again, don't worry too much about section 2.1 and 2.2 for this chapter. Start with 2.3 and work your way through the rest of it, because that's really where we're going to focus is our acids and bases and our inorganic versus organic molecules. Okay. I hope you guys have enjoyed your long weekend, and I will maybe see you all in class. Have a good day.